Hello everyone, welcome back to our lectures. Emeka Udenze is speaking once again, and today we'll be talking about enzymes, inhibition and control. The picture I drew here is a picture of non-competitive enzyme inhibition, and this is part of what we're going to be talking about as we move into this lecture. As usual, we're going to be looking into the learning objectives. We have the following learning objectives to cover in this class. Number one, we'll describe the types of enzyme inhibition. Two, we're going to describe different categories or different levels of enzyme regulation or control. We can call it regulation or control, which include Zymogens, allosteric, and genetic control. And finally, we're going to be discussing the importance of blood serum enzyme levels in diagnosis and prognosis of diseases. I have some acknowledgement to make. Now, most of these beautiful pictures you see in this class, I've done them with a lot of open resources, a lot of resources that have been great. And I give credit to some of those ones. The credit to the software tools used in making this video include the BioRender, the BioVia, Screencast-O-Matic, and then the OpenBoard. I've used all these ones to make this video, And most of these are free, and some of them have some paid. This is completely free, and then this is completely free as well. But this one is free, but if you want your video longer, you can subscribe for other paid versions, including BioRender as well. All right, let's just look at the quote as usual, as I do in my class always. In a popular face in the United States, LeBron James is a 4-time National Basketball Association champion. He's currently a player for Los Angeles Lakers, and they are the current defending champions of the NBA. He is, like I said, he's a 4-time winner, and he is a social justice advocate, someone who spends his time and money to make sure that social justice issues are addressed in the society. Let's hear what he has to say. He said, "Don't be afraid of failure. This is the way to succeed." LeBron James. He has been to so many finals, about 10 of them, he has only won 4. So he understands what it means to keep trying. And if you keep trying and failing, that is the only way to succeed, to learn and move on and then succeed. Let us immediately start up the lecture. Enzyme inhibitors. Enzyme inhibitors are substances that bind to an enzyme and prevent it from performing its catalytic function. Remember, the main role of enzyme is to perform the catalytic function. But when you prevent it from performing that function, that enzyme has been damaged. It is not living up to its calling. There are 2 types of inhibitors, the irreversible inhibitors and the reversible one. Of course, the irreversible ones are those ones that cannot be undone. The moment they occur, it damages the enzyme completely and the enzyme will not function anymore. Whereas the reversible ones can be undone or reversed. That's exactly what it means. Now, what is the importance? What is the significance of enzyme inhibition? Now, the mechanism of action of most poisons and pharmaceutical products like drugs, medicines, is to inhibit a specific enzyme in a biochemical pathway. And if they inhibit that particular enzyme, what do? They either decrease or even stop the catalytic process. So if you're taking an antibiotic, what an antibiotic is doing is to stop a particular enzyme in the biochemical process of the bacteria that makes it to survive. And if it dies, you get well, the drug is working. Now, the drug or medicine is actually working. Now, some substances found in cells inhibit enzyme catalyzed reaction. Yeah, some substances that are normally found in a cell will inhibit some enzyme catalyzed reactions so that those reactions do not proceed indiscriminately. What does that do? It affords the cell a natural means of internal regulation of metabolic reaction, because metabolic reaction need to be regulated. If they are not regulated and this reaction goes out of whack, bad things begin to happen in the body. Now, so let's look at irreversible inhibitors. Irreversible inhibitors covalently bind to specific functional groups at the active site of the enzyme and render it inactive. In fact, what they basically do is to kill the enzyme. That's exactly what they do. They kill the enzyme and it doesn't work at all, damages the enzyme. And they include the following poisons. Poisons such as cyanide. Cyanide is a very dangerous poison that acts very rapidly and even begin to shut down the cellular respiratory process. Other ones include heavy metals such as lead and mercury. We've talked about the mechanism of action of lead and mercury when we looked at the thiols. What we said is that they form this permanent bond with the thiol functional group of cysteine, and as a result, also damage the enzyme and not allow it to function properly. And the last group there is the antibiotic. Antibiotics such as penicillin and sulfur drugs inhibit enzymes that are essential for the survival of the bacteria in the host organism, in humans or animals. Now, what does penicillin do? Penicillin specifically interferes with an enzyme that is called the transpeptidase. Now, this transpeptidase is an enzyme that is essential for the bacterial cell wall synthesis. What does it do? When this particular enzyme is interfered or inhibited, it prevents the bacteria from making strong cell wall for protection, and ultimately leads to its death. And when it dies, we say the penicillin medicine is actually working and has done its job. So in this case, the idea is to kill or shut down the bacteria from causing harm in the body. Okay, we now go to the next thing. Cyanide poisoning. Let's look at it. Cyanide ion poisoning. Cyanide is an extremely toxic substance and acts very rapidly or very fast. What it actually does is that it interferes with the functioning of an ion-containing enzyme. That ion-containing enzyme is the cytochrome oxidase. Very important enzyme in the respiratory process. So what does it do? It forms a stable complex and does not allow the enzyme to function properly. And this results in the cessation or the stoppage of cellular respiration. You know what it means? When cellular respiration stops, it causes death even in a matter of minutes. It is very, very dangerous and needs to be treated as an emergency. Now, particularly when exposed in large quantity. Now, so this is cytochrome oxidase. Cytochrome oxidase has an ion as three ion as the prosthetic group in a normal enzyme. Now look at what it does. The cyanide ion comes here and binds to it to form this permanent compound. So look at what it does. It comes here and binds with it to form this permanent complex, this stable complex. Now this is the active enzyme. At this place, this enzyme is inactive. And when this thing happens, it becomes a serious danger that needs an emergency action. How is it treated? It is treated with sodium thiosulfate. Sodium thiosulfate is an antidote that converts cyanide ion to sodium thiocyanate, or you can say thiocyanate ion, which does not bind to the ion of the cytochrome oxidase. So what it basically does is that it takes all the cyanide that the person has in the system and convert them to thiocyanate. So this is the cyanide ion. This is the cyanide ion. Now combines with the thiocyanate, sodium thiocyanate here, to form, cyanide ion combines with the sodium thiosulfate to form thiocyanate, and then the sulfide. Then thiocyanate is very soluble and is now excreted by the kidneys. And that is how this is actually treated. Like again, I said, sodium thiocyanate is an antidote for the treatment of cyanide poisoning. Now let's start talking about reversible inhibitors. Reversible inhibitors, just like the name implies, are inhibitors that reversibly bind to an enzyme. They can be reversed or they can be undone by shifting the established equilibrium between the enzyme and the inhibitor. Look at what happens. I here is the inhibitor, E is the enzyme. So E reacts with I to form this. Remember, this guy cannot go and release the enzyme and form the product. No, this is not possible. This will not happen. It doesn't happen here at all. This doesn't happen. Therefore, when you have this, you're not expecting any product to form, because in a normal reaction, an enzyme will have to combine with the substrate is S to form the enzyme substrate. Remember, the formation of the enzyme substrate complex is the rate limiting step here. This is what you want to form. And then this guy is now going to break down to release enzyme plus the product. So this is the most important step that need to form. You want this step to form. If this step doesn't form, the enzyme substrate complex, it doesn't change to the enzyme transition state complex, which eventually forms the product and releases the enzyme when you have this. So there are two of them we're going to be talking about here. They include the competitive inhibitors and the non-competitive inhibitors. The competitive inhibitors, look at the word competitive, they compete with the substrate for the binding on the active site. Now, this type of inhibitors are substrate analogs or lookalikes. What they do is to compete with the enzyme for that small tiny portion of the active site where catalysis takes place. Whereas the non-competitive inhibitors, what do? They bind to another location other than the active site. They leave the substrate to take the place of the active site, whereas they look for another location on the active site. And this, like I said, the enzyme, the inhibitor does not bind the active site of the enzyme. So, and this type of, the enzymes involved, they are usually called what we call the allosteric enzymes, as we're going to see in this lecture as we move on. So let's look at competitive inhibitors. They have molecular structures that are similar to the normal substrate of the enzyme. So therefore, we call them what? Substrate analogs or lookalikes. They look exactly like the substrate. So they are mimicking the substrate. That's exactly what they do. Now, the binding of these inhibitors prevents the formation of the enzyme substrate complex, which is an important intermediate for the product to form. Now, this type of inhibition can be reversed since it's a competition. Remember, in competition, anybody who takes over first or who is there faster takes control. So this can be reversed by increasing the concentration of the substrate. So if you increase the substrate concentration, this type of inhibition is reversed. That's why we call it a competitive reversible inhibition. All right, so let's see what happens here. This picture, this is a beautiful picture that I actually designed myself from BioRender 2. This is the enzyme. If you look at this enzyme now, this is the enzyme. This is the competitive inhibition. This enzyme has a ridge. This ridge looks like a rectangular shape. Now look at the normal substrate that has this shape to bind here. Now look at the inhibitor. This is the inhibitor. It does not look like this at all, but it looks like, okay, let me work that back. Now this inhibitor is not the substrate, but it looks like the substrate only in this point that it wants to bind on this guy. So look at what happens. If the inhibitor gets to the enzyme first, it takes the place of this and binds. And when it binds, it prevents the substrate from binding. And when it does prevent the substrate from binding, the enzyme substrate complex does not form. However, if you want to reverse this, you have to increase the concentration of this so that this guy will go back and push this away and takes over this. So here, they're competing for someone who's going to take over this space because both of them have the same shape that they will use in binding to the enzyme. So in equilibrium, look at what actually happens. Remember, in a normal reaction, enzyme want to bind to a substrate to form the enzyme. Okay, that is an equilibrium reaction. Usually that's how we write it. To form the enzyme substrate complex. And this will actually break down to form enzyme plus product. However, look at what it does. Now this guy goes here to bind this guy. If he binds, he forms enzyme plus I. That's what happens. It forms this. And if the equilibrium all moves over to the formation of this particular reaction is not going to take place. So what do? Is to increase the concentration of this and make sure that it ties up this enzyme to push the equilibrium towards the right for the formation of this guy. That's exactly the mechanism of action of competitive inhibitors. All right, like I said again, the most important thing to take away here is that they compete for the active site, and the inhibitor here is a lookalike or substrate analog of what? The substrate. Okay. The non-competitive inhibitors. Now, like I said earlier, they do not look like the enzyme's substrate at all. They don't look like that. The interaction between the enzyme and the inhibitor changes the conformation or the three-dimensional structure of the enzyme and its active site. What it actually does is that it binds in another site, right? Quite all right. It binds in another site. However, that binding, what does it do? It changes the three-dimensional structure of the active site, that the active site is no more able to accept the substrate, despite the fact that it is not occupied. So substrate, as a result of this, substrate is improperly bound or not bound at all to the active site. It restricts the catalytic group of the active site from involving in catalysis. That's exactly what it does. It restricts the catalytic group of the active site from involving in catalysis and prevents the proper formation of the enzyme substrate complex. However, if it cannot form the ES complex, product cannot form at all. Catalysis is not taking place. So substrate concentration does not affect the inhibitor action. So the increase or decrease in substrate concentration does not actually overcome this inhibition. The only way it can be overcome is by reducing the concentration of the inhibitor itself. Let's see what happens here. Now, if you look at this structure, this is the inhibitor, this is the substrate. Now look at what happens. The inhibitor can fit in here because it has the exact shape for this. This is like a rectangular shape. It can fit in easily. Look at this triangular shape here, and this one can fit in easily. And it's expected that both of them will fit in. Look at what happens here. Now, the moment the inhibitor comes here and binds, it changes the conformation of this guy. Look at this, looks like a triangle. This looks like a triangular shape. Let me call it triangular shape. To a rectangular shape. That's what it does. Because the binding changes the conformation at this point. From triangular shape to rectangular shape. Let me use a different color. Changes it from this triangular shape to rectangular shape. And look at it, the substrate has a triangular shape, therefore it cannot bind there. So what it basically does is that it does not take the place of enzyme in the active site. The active site, remember, this is actually the active site. This is the active site. The active site is there. What it simply does is that the binding changes the conformation of active site, that it is not able to accommodate the what? The substrate anymore. That's exactly the mechanism of action of this enzyme. So if you write the equilibrium like I did for the other one, remember the normal one, E will react with S to form the ES complex. And ES complex will break into E plus product. This is the normal reaction. What does it do? It comes here, binds with this guy to form enzyme inhibitor complex. Of course, the enzyme inhibitor complex will not give you the product. By tying up this to this equilibrium this way, you're not going to be forming this guy at all. It can also do what again? Look at another thing tricky about this guy. Now, it can also bind with the substrate to also form the ESI complex. Yes, at times. But here it's not improperly bound. Even when it is bound, it is not improperly bound. It can also come as well and bind on this guy. It can bind on this guy too to form the ESI. Now, in all this equilibrium, none of them will result to the formation of the product and releasing the enzyme, and therefore catalysis is actually inhibited. We now go to the next objective, regulation of enzyme activities. Now, I told you there are three categories of regulation of enzyme activities we're going to be talking about. We're going to talk about the Zymogens, we're talking about the allosteric enzyme, and we'll talk about the genetic control. So Zymogens or proenzymes are inactive precursors of enzymes. Now, some enzymes that can degrade the internal structures of the cell are stored as inactive Zymogens. Now, some of these enzymes are very, very active. If you store them in the cell when they are not needed, they begin to corrode the cell. They begin to destroy the cell. So you don't want them to degrade the cell. So they are naturally stored as inactive enzymes called Zymogens or prohormone. They are only released when they are what? Required. When the cell needs them, at cellular need, that's when they are released. And after release, they are now activated at their site of reaction. At their site of biochemical reaction is where they are activated. How are they activated? That the activation involves the cleavage or hydrolysis of one or more peptide bond in the Zymogen. The Zymogen is usually a polypeptide. So if you break out a few peptides from it, it becomes active. And good examples are mainly, we see them mainly, the common examples that occur in other places, but the common examples are the digestive enzyme, which include the trypsinogen, the pepsinogen, and the chymotrypsinogen. Remember, the trypsinogen is converted to the trypsin, which is the active enzyme. The pepsinogen is converted to the pepsin, which is the active enzyme. The chymotrypsinogen is converted to chymotrypsin, which is the active enzyme. A good example here is that trypsinogen is the inactive one. Now, it is an enzyme now chops off some hexapeptide to convert it to what? Trypsin, which is the active enzyme. It is the trypsin that now gets involved in catalytic action. Now we see the second level of enzyme activity, enzyme regulation. The second one is allosteric regulation. And these enzymes, we call them allosteric enzymes. You see, allosteric regulation refers to control mechanism involving allosteric enzyme. What are allosteric enzymes? Allosteric enzymes are enzymes that change the conformation of their quaternary structure. Remember, an enzyme that has a quaternary structure has more than one subunit. Remember that. Any enzyme that has a quaternary structure has more than, okay, let me write that well. We talked about this in proteins. Has more than, that than is not coming out well. More than one subunit. Or you say more than one polypeptide chain. So look at what happens again. I said these allosteric enzymes are enzymes that change the conformation of the quaternary structures upon binding of modulators or effectors. So upon binding of some substances, they change their quaternary structures. And this results to the change in their what? Three-dimensional structure and hence their function. Remember when we talked about the protein's structure and function, we said a change in three-dimensional structure of protein, which includes enzyme, will always change their functions. And what are modulators? Modulators are compounds that bind to a site other than the active site. Now look at what happens here. Allosteric enzymes have more than one active site, just like the non-competitive inhibitors. I told you, allosteric enzymes are non-competitive, operate by non-competitive inhibition for their regulation. Look at what happens. Those enzymes that are involved there have more than one active site. They have the catalytic site and the regulatory site, which we call the allosteric site. So now, these modulators we're talking about are substances that bind to that allosteric site, that is not the active site, and cause them to change the conformation of the active site and hence affect their catalytic activity. So modulators are compounds that bind to the site other than the allosteric enzyme's active site and alter its catalytic activity. Now, they can do it in two ways. They can either increase this activity, we call them activators, or positive modulators. We say positive modulators. Or they decrease this activity, we call them inhibitors, or negative, you can say negative modulators. That is exactly what they are. And they do this, and there's another important thing we're going to link to this. Now, okay, before I talk about feedback inhibition, let me give you an example of what I mean by this enzyme. Now, if this is an enzyme, let me make an illustration. An enzyme has two polypeptide units. Let me make a small sketch there. It has two polypeptide chains, one facing this way and another one facing this way. These are two polypeptide chains. Look at each of them have a groove. The groove on this side, let's assume this is the active side. And this one now is what? The regulatory side. Just like, look at it, just like what we have in this inhibition. They have two sides. One side is the active, and the other side is the regulatory side, because it is what determines what is going to happen at the active side. So that's exactly what happens. Remember, these two are polypeptide. Okay, let me use a different color to make it clear. Those two are These are the polypeptide chains of subunits. Polypeptide chains Now look at what actually happens here is that if a modulator binds on the regulatory site or allosteric site, it will change the conformation of this site and make it in such a way that this site is not able to react well with the substrate and therefore slows the catalytic ability of that enzyme. And this particular operation is applied in the feedback inhibition of metabolic pathway. What is feedback inhibition? A feedback inhibition is a process in which the product of a sequence of enzyme catalyzed reactions inhibit the earlier step of the reaction. Now, if an enzyme is catalyzing a reaction and producing a lot of maybe an end product, if that end product begins to pile up in high concentration, that the body doesn't need too much of it, what does it do? That particular product is going to go back and begin to tell the enzyme, stop my production. That is what we call it feedback inhibition. Feedback inhibition. And so we're going to see in the next page. So look at what happens now. Before I read this, let me tell you what happens in feedback. In feedback inhibition, now this is the original substrate. And this substrate has a series of enzymes. Remember, a biochemical pathway is a pathway that involves a series of enzymes that catalyzes an original substrate until you get a final product. That's what we call a biochemical pathway. Now look at what happens. It starts from the original product, acted upon by enzyme 1. Enzyme 1 converts this original substrate to product 1. Enzyme 2 converts this product to product 2. Enzyme 3 comes in until you get the final product of this pathway. Now, at some point in this pathway, the final product concentration begins to increase and pile up. That it is now too much that the cells do not need it in excess. What happens? This particular final product is going to now loop over and go and bind on the allosteric site of this enzyme 1 and slow it down so that it does not keep catalyzing the formation of this one, which will result in the formation of itself ultimately at the end of the day. This type of inhibition is what we call feedback inhibition. An example of examples of allosteric enzymes involved in feedback include, there are so many of them, include threonine deaminase, TDNase, and aspartate transcarbomoylase, we call it the ATKs. Now, both of these enzymes catalyze series of reactions in which the final product bind to the allosteric site of the earliest enzyme in the pathway. What happens? The product here exerts their inhibitory effect on that enzyme. In this case, what do? The reaction pathway slows down as the concentration of that product increases. And the implication here is that the product is never made in excess, providing a perfect internal means of regulation of these enzymes involved. So the final categories of regulation of enzyme activity we're going to be talking about is genetic control. This one is actually very interesting. I usually tell students this. You see, genetic control, simply remember, every protein or enzyme is synthesized by the gene from the instruction of the DNA as encoded in the genetic code. So the synthesis of all protein, including enzyme, is under genetic control. That is actually what helps them. It's a translation process. The DNA is replicated, and then after replication, the RNA is produced in transcription, and the RNA now directs the synthesis. The RNA now directs the synthesis from the instruction of the DNA. The RNA directs the synthesis of the protein in the translation process. In fact, this is actually the central dogma of biology, of information transfer in living organisms. Now, so that leads us to what is called enzyme induction. What is enzyme induction? Enzyme induction is the synthesis of an enzyme in response to the cellular need. Synthesis of an enzyme in response to cellular need. It allows organisms to adapt to environmental changes, rather. It allows organisms, cells, to adapt to environmental changes. So if a cell stays and something changes, it has to adapt to it because it's okay, something is unusual about the concentration of a particular product. Therefore, what the cell will do is to give signal for the production of an enzyme that will metabolize that particular substance produced in excess. So the synthesis, a good example is what we're going to talk about. The two good examples I'm going to be telling you is the synthesis of alcohol dehydrogenase, ADH, and beta-galactosidase. Now, alcohol dehydrogenase is an enzyme that metabolizes or that converts alcohol or ethanol to acetaldehyde. Then so look at what happens. Alcohol is converted by alcohol dehydrogenase to acetaldehyde. Then acetaldehyde is now converted to acetate by aldehyde dehydrogenase. This acetate is now converted to acetyl-CoA by acetyl-CoA synthetase. Look at what happens here. Now, acetyl-CoA, the moment you form acetyl-CoA, you have three fates. It has a lot of fates. It can get into tricarboxylic acid cycle and used for energy. It can be used to form fatty acid in the process of lipogenesis. It can also be used to form proteins, and it can also be used to form ketone bodies. Mainly, it channels mainly to formation of fatty acid and ketone bodies, both of which you don't want to have a lot of them in the body. So the metabolism of alcohol happens like this. Now, what is the scenario? Let me tell you the scenario. This is the most interesting part. So, have you wondered why some people can take a lot of alcohol, a lot of alcoholic beverages without getting knocked out? And some other individual will take it and just get knocked out immediately. Do you know the reason? It is because of the induction of alcohol dehydrogenase. What does it mean? Now, someone who is already used to drinking a lot, will have higher concentration of alcohol dehydrogenase because the cellular environment is already adapted or used to seeing a lot of concentration of ethanol in the blood system. Therefore, this enzyme will always be there to take care of the alcohol. Therefore, minor or little consumption of alcohol is not going to knock the person off. The person is not going to be intoxicated by few drinks, by few cups of alcoholic beverage. However, for someone who has not had an alcoholic beverage before, or is not used to having it in period, like you've not had it for the last 1 year, and all of a sudden you take a bottle of alcohol, you'll be knocked out because your body does not have enough alcohol dehydrogenase in the system to begin to, in the cells rather, to begin to metabolize that excess alcohol you are pushing into the body. So this is an enzyme induction. This is the enzyme induction of alcohol dehydrogenase. The same thing happens in beta-galactosidase. The beta-galactosidase is the enzyme that does what? That catalyzes the breakdown of lactose, milk, into galactose and glucose. Now, have you wondered why some people have lactose intolerance? Someone who has lactose intolerance cannot make enough galactosidase to break down lactose. And this happens particularly, some individuals are born in some part of the world whereby they did not take enough milk. They didn't have enough milk when they were growing up as a child. And all of a sudden, when they're grown, they take milk, they begin to have stomach upset. The stomach upset they're having is because the body or the system does not have enough beta-galactosidase that will break down the lactose into glucose and galactose. And that particular scenario, that particular biochemical scenario is what we call enzyme induction. And it is because of the genetic control of enzyme activity. And then having said that, we now go to the last thing, which is enzyme in clinical diagnosis. What is the application of enzymes, biochemical enzyme level in clinical diagnosis? You see, changes in blood serum enzymes. Okay, let me use my usual color. Changes in blood serum concentration of specific enzymes is used as a marker to do three things. There are three things we use it for. Number one, to detect cell damage and uncontrolled cell proliferation. In the case of cancer, when the cells go whack, begin to reproduce indiscriminately, that's what you can use this. You can use these enzymes as markers. Two, you can also use it to suggest the site of damage or site of cancer or excess cell proliferation. They can also be used to estimate the degree of cell damage. And how does this happen? This is done by determining the magnitude of the increase in the serum concentration of the enzymes above the normal level. Now, these enzymes in the bloodstream have a normal level that they're supposed to be when the body is healthy, when the cells are healthy. However, when the cells began to be injured as a result of pathogen, as a result of any pathological conditions, the levels of these enzymes will begin to go high. So the early detection of these levels of enzyme can be used as a serious marker in predicting what could happen in the near future. And that will inform the proper treatment of that particular pathological condition. So the measurement of enzyme concentrations in serum has become a major diagnostic tool in the diagnosis and prognosis of diseases of the heart, the liver, the pancreas, the prostate, the bones, and so many organs of the body. So when a particular organ is marked by a particular enzyme, detecting that enzyme in the body in an unusual concentration is a potential sign of a particular pathology tied to that particular organ. And in the next page, we're going to be looking at, which is fortunately our last page, we're going to be looking at some enzymes and their particular markers in disease conditions. So the first column is the enzyme, the second column is the disease or pathological condition. You can say pathological condition as well if you want to use that word. So let's see what happens. So these are serum enzyme levels and disease. Alanine transferase or ALT. When you detect high levels of alanine transferase in the bloodstream, in the blood serum by diagnosis, it's usually a sign of either hepatitis or some kind of liver damage or other liver diseases. Aspartate transaminase can also be AST, it can also be used for hepatitis and heart attack. Acid phosphatase is used for prostate cancer, and this is actually very important. And people who are up to 40 years need, particularly the males, will always go for prostate cancer screening. And this actually shows up very much early as an important marker of prostate cancer in the near future. Amylase is usually used for pancreatic disease, either pancreatitis or other type of pancreatic cancer. Alkaline phosphatase is used particularly for bone and some type of liver diseases. Lactate dehydrogenase particularly is a good marker both for heart and liver disease. It's more specific for the heart particularly. And then creatinine kinase is usually used as a marker in heart attack and even at some point in kidney diseases. I didn't put that down. And finally, lipase. Lipase is also used as a marker in pancreatitis. And having said this, we've come to the end of this lecture. Thank you for listening. Bye.